Thanks for joining us for another RRP hosted research webinar. I'm Katrine Erickson. I'm the Executive Director of the RUNX1 Research Program. I think that most of you joining us, although I can't see your faces or your names at the moment, probably are already familiar with RRP, but just in case any of you are new to who we are, we're a nonprofit organization that's dedicated to supporting patients with RUNX1 familial platelet disorder, which you may already know is a genetic disease caused by mutations in RUNX1. And unfortunately, one of the most concerning issues for patients is their very high risk of hematologic malignancies. And so we were founded by a patient family that wanted to establish one single goal for our organization, and that is to prevent the onset of hematologic malignancies in individuals with RUNX1 FPD, and of course, also improve their overall quality of life. Our work not only includes funding research, and that's what we're going to discuss later today is one of the funded research projects, but we also do spend a lot of time building and supporting not only our patient community, but also our medical and research community, and then really facilitate collaborations between those communities so that we really work in concert together. And again, that work is all focused on this single goal, which is to discover a blood cancer interception. Our impact in terms of research dollars has now reached over 13 million. And that's really only been possible thanks to a number of different partners we've had along the way, which include the Alex's Lemonade Stand Foundation, the Leukemia Lymphoma Society, the Mark Foundation for Cancer Research, the Edward P. Evans Foundation, and I'm missing someone, and now the American Cancer Society. So we've had some wonderful partnerships and friends, and that has helped us really fund quite a bit of research. If you're currently conducting RUNX1 FPD research or RUNX1 relevant research and are interested in presenting at one of these webinars, please do reach out to me. These are really ad hoc. When I hear that someone's got a new story that they're ready and willing to share, I reach out to them and say, please join us for a research webinar. But if you have something that you'd like to share and I don't know about it, please let me know. We'd love to host you. Also, if you're a researcher and you're interested in applying for one of our grant programs, we have usually at least one grant mechanism available each year, but sometimes it's more. And so I highly encourage you to check back on our website or sign up for our newsletter so that you keep apprised of when we have these open grant mechanisms. And I can tell you we'll have one with Alex's Lemonade Stand Foundation that will be due at the end of December. We'll probably announce in the early summer. So keep your eye out for that. That's called the RARE grant program. And then we're hoping to announce a second grant mechanism this year as well. So again, please reach out to me or just make sure you read your emails and read our newsletter, and you'll be well informed. Before I introduce our speaker today, just a quick reminder that we're in a webinar format. So please do ask your questions. You're more than welcome to put them in at any point in the presentation. They just sort of create a list for me in the Q&A icon, and I will go ahead and ask the questions at the end for you on your behalf. All right, so without further ado, I'd like to introduce Dr. Kristy Stengel, who is an assistant professor in the Department of Cell Biology at Albert Einstein Medical School in New York. Dr. Stengel's research focus is on transcription factors and how they are influenced by and exert influence over the chromatin landscape, how multiple sequence-specific transcription factors cooperate to fine-tune gene expression, and many other fundamental questions that she's asking around the role and function of transcription factors. And she uses a chemical genetic approach to rapidly degrade endogenous transcription factor proteins to provide those answers to those critical questions. Most importantly, Dr. Stengel is a RRP ALSF early career investigator. And as I mentioned earlier, the work that she's sharing today with us is in large part from her funded project. The title of her talk is Transcription Factor Associations Fine-Tune the RUNX1 Gene Network to Confer Regulatory Logic to Hematopoietic Gene Expression, or gene expression programs. We are delighted that you're here with us, Kristy, and I'm looking forward to hearing your presentation. Over to you.
All right, great. So can you hear me and can you see my slides? Are we good there?
We're good.
Yes. Okay. So thanks, Katrine, so much for having me, and thanks to RRP for being really the first funder of my new lab, fledgling lab at Einstein. We're really appreciative of the support, and I think particularly in this day and age, all of us in science are very appreciative of the great work these foundations do supporting our research. So what I want to talk about today is our work, our attempts to study RUNX1 regulated gene expression. And I know with this audience, there's no need to introduce RUNX1. So what I thought I would do with my introduction is spend time talking about how we think about transcription factors and their involvement in gene regulation, and how we approach the study of transcription factors, and in this instance, RUNX1. I'll talk about how we've applied these chemical genetic approaches that Katrina alluded to kind of understand, how RUNX1 is working to directly alter gene expression programs. And then I'll touch on our work looking at the contribution of other sequence-specific transcription factors, which converge to either antagonize or cooperate with RUNX1 in the regulation of its gene network. And then if I have time at the end, I put in a couple teasers of some projects that are ongoing in the lab that given the tight community, RUNX1 community, I thought I may like to share. Okay, let's see. So I give a lecture on transcription factors here at Einstein, and this is one of the first slides that I always show, because when we stop and think about how essentially every cell in our body has the exact same DNA content encoding the exact same genes, and yet there's such a diversity of phenotype and function of these cells, we really understand that it's really the proteins that regulate which genes are expressed and when they're expressed that really underlie these profound differences in cell phenotype and function. And those proteins that control, when and where other genes are expressed are transcription factors. And we think of these as really the master regulators of cell identity and cell fate. At last check, and this may have changed, there was an estimated 1,600 transcription factors encoded in the human genome, which means roughly 8% of human genes encode for proteins that control the expression of other genes. And I think this just underlies the profound importance of this class of proteins in regulating cell development and function. What I also think underscores the profound importance of this class of proteins is the sheer frequency with which they're mutated in cancer. And I think we all appreciate that altered gene expression really is a hallmark of all cancers. And particularly in the context of myeloid malignancies, we see oftentimes either chromatin regulators or transcription factors themselves are highly, highly mutated. And this can come in the form of translocations involving these transcription factors, of which a particular translocation involving RUNX1 that fuses it to a co-repressor protein called RUNX1T1, previously known as MTG8 or ETO, leads to a dominant repression of the RUNX1 gene network. And again, this is one of the most frequently recurring translocations in AML, and really a protein that we've studied in the past. But also, mutations in the transcription factors themselves. Oftentimes, in the case of RUNX or CBP alpha that are referenced here, these are inactivating mutations. But even the tumor suppressors that are referenced in this figure actually, encode transcriptional proteins. And so I think this just highlights the importance of proper gene control for normal function, and how when things go wrong, it can lead to cancer development. Now, in addition to just the sheer number of these mutations, we also know from studies like the cancer dependency map study that, transcription is a profound vulnerability in many of these diseases. But by and large, we've failed to capitalize on that ability, because oftentimes these transcriptional regulators are what we consider undruggable targets, particularly the transcription factors, due to the highly disordered nature of the proteins. And so really, the way my group approaches transcription factors is we think the more we know mechanistically about how they work, the more likely we'll be to identify perhaps novel nodes for therapeutic intervention in these transcription factor mutated diseases. Okay, and so this is really kind of at the crux of how our group thinks about transcription factors and how we go about studying transcription factors. So transcription is a highly dynamic process. It's regulated in the order of minutes to hours, and that's important because cells need to be able to rewire gene expression networks to respond to a variety of intercellular and extracellular cues. So they really have to be nimble and agile in their ability to turn genes on and turn genes off. In contrast, historically, kind of the methods and the technologies that we've had at our disposal to study these highly dynamic proteins tend to be pretty slow to eliminate the transcription factor protein function. And, we've largely because, as I mentioned, these are kind of undruggable targets, we've been left with genetic approaches to eliminate transcription factor protein. And this kind of is at the far end of these slow approaches that have been utilized to study transcription factors. Genetic knockouts, such as animal models, which are fantastic models for understanding what are the physiological consequences of a loss of a transcription factor on development, on interaction with the microenvironment. There are numerous questions that simply can't be addressed in other systems. And these animal models have been really, really useful in our understanding of RUNX1 as well as many other transcription factors. The problem or the limitation with these models comes when we try to understand the molecular events that led to these changing phenotypes. And that's because when you eliminate a transcription factor protein, again, they're highly dynamic and transcription is changing in minutes to hours, you get a direct effect on the genes that transcription factor regulates. But in the days to weeks that these tissues develop in the absence of this transcription factor, these direct targets will regulate. Some of them are transcription factors that will regulate their own target gene expression. Some of them are signaling molecules that ultimately impinge on gene expression networks. And so what you have by the time you're able to assay changes in gene expression are really a combination of both direct effects on gene expression as well as secondary, tertiary, and compensatory gene expression changes that are really a reflection of altering cell states and phenotypes and more of a description of those phenotypes than the underlying cause. And so at the other extreme of this kind of range of kinetic resolution that we have for studying transcription factor is small molecules. So small molecules are able to inactivate protein function very, very rapidly. And so this is really the direction we would like to go to eliminate the confusion and the confounding identification of non-direct gene targets to just get this first layer of gene regulation. Now we know for transcriptional proteins for which we have existing small molecule inhibitors that we can see changes in gene expression very quickly. And so here I'm just showing an example of a small molecule you probably all heard of called JQ1. That's a bromodomain inhibitor. This inhibits a protein that's involved in the release of RNA polymerase pausing into productive elongation. Here in red, all you need to know is the signal is polymerase transcribing across this KIT locus. And when you treat with this JQ1 molecule for just 15 minutes, you lose the release of that pause polymerase into productive elongation. And you see a rapidly diminishing signal at the 5' end of the gene. At the same time, at the 3' end of the gene, those polymerase molecules that had already escaped are now exiting the locus, and there's not yet an effect on the polymerase levels here. But by 30 minutes, the KIT locus is maximally silenced. And so what that tells us is that these small molecules have the ability to induce rapid changes in gene transcription, and really in the order of minutes to hours, the direct effects of these small molecules can be seen. And I just wanted to show if we use small molecules and wait even longer, not too long, just 8 hours to 24 hours. What we see is if you look at gene expression, we really see waves of changes happening. So we think even waiting a day after you've perturbed these transcription factors, you're starting to see these really indirect effects on gene expression. And so what we want to do is kind of narrow the time frame for studying transcription factor inactivation from days to weeks using these knockout models to minutes to hours using small molecules. But as I mentioned, transcription factors are largely undruggable. And so the way that we can get around this is using technology for targeted protein degradation. And so what my group does is we use CRISPR-based approaches to engineer endogenous transcription factor loci with this small peptide tag, FKBP12 mutant tag, that then renders the protein sensitive to a small molecule, in this case called DTAG. So this is a PROTAC, it's a heterobifunctional molecule. One side of that molecule binds to the transcription factor with this tag, the second binds an E3 ligase. And by just inducing ternary complex formation, you get rapid ubiquitination and subsequent degradation of the transcription factor you're studying. So we've applied this approach to multiple transcriptional regulators. As I alluded to previously, the first attempt we did to describe a transcription factor using this technology was actually this RUNX1 EDO fusion protein. And so this is really how we came to the RUNX1 field. We had this first study studying this fusion protein, and then we wanted to really dive in and ask, could we apply this technology to now understand what the wild type RUNX1 protein was doing? Okay, so I'm just going to jump right into the data now. So all of you know that RUNX, there are three annotated isoforms of RUNX, RUNX A, B, and C. And this is always one of the first things we think about is the structure of a locus when we're thinking about adding one of these Degron tags. So RUNX 1B and C share the same 3' last exon. And so they can be tagged, but with a C-terminal tag. So C-terminal tag would eliminate both RUNX1C and RUNX1B protein. However, it would not eliminate RUNX1A. And so that is the caveat of the approach we've taken. That being said, the HEL model system in which we've used for at least our initial studies, we see by RNA-seq and Western blot, very little evidence that RUNX1A is expressed at any appreciable level. So we think by introducing a C-terminal tag, we're going to get most of the RUNX1 protein. So we use this CRISPR-based approach to integrate this FKBP-based Degron tag, and we also incorporate a 2xHA tag for biochemical analysis. We also include a P2A ribosomal skipping sequence and a fluorescent protein that allows us to sort for successful integration of the tag into the endogenous locus. So the only way the fluorescent protein will become expressed, because it doesn't have its own promoter driving its expression, is if it's integrated in frame into an open reading frame that is actively transcribed. And so we use this approach to then sort for the edited cells, and you can see in these HEL cells, RUNX now migrates more slowly on the gel, consistent with the incorporation of this Degron tag, and we can detect the RUNX1 protein using an antibody against HA. One of the first things we do, and one of the caveats that we will always acknowledge of the system, is we are adding a tag to an endogenous protein that has the ability to impact its function. So we will look at RNA-seq generated from our cell line before and after tag, no addition of any molecule to trigger degradation, and ask, does the addition of the tag alone significantly influence gene expression globally? And we see no evidence that the addition of the tag to RUNX1 resulted in global changes in gene expression. We can then work out the conditions for degrading RUNX1 through the addition of the DTAG PROTAC small molecule. And you can see within really largely by 1 hour, almost all the RUNX1 protein is gone, and it's completely gone by 2 hours. So this is going to provide us, again, with that kinetic resolution that we think we need to really hone in on these direct effects on gene expression. One of the first things we did was just ask, does degradation of RUNX1 in this model have any phenotypic consequences? So we could take parental cell lines and treat them with this DTAG degrader, nothing happens to cell growth. If we take the RUNX1 tag cells and treat them with this degrader, we do see that the cells grow slower, though I will admit it's a pretty subtle reduction in cell growth. And it's really due to, I'm not showing this data here, but a lengthening of G1 that seems to take roughly between 24 to 48 hours to manifest. So this isn't an immediate effect of RUNX depletion on cell cycle and cell growth. It takes a little while, and you ultimately have a reduction of cells in S phase. We can also use this model to trigger differentiation towards a megakaryocyte-like cell fate. So obviously it's a cell line, so it's not perfect, but we can treat cells with PMA and kind of induce them towards the CD41, CD42 double positive cell fate. And when we do that, we see that degradation of RUNX results in a complete halting of that progression to the double positive fate, suggesting this may be a reasonable model to look at RUNX1 transcriptional targets given, the phenotypic consequences that we're seeing. Okay, so I want to talk a little bit about how we measure changes in gene expression, because I think the ability to inactivate the protein quickly is only one side of the coin. The other side is that we have to measure changes in transcription very quickly. And we do this by measuring nascent transcripts specifically. And we use a technique called ProSeq. This is essentially a nuclear run-on performed in the presence of biotinylated NTPs, such that when the polymerase incorporates one of these bio-NTPs, it's actually chain terminating and the polymerase will stall. And so we can purify those nascent transcripts based on the biotin-streptavidin interaction. And using that, we sequence from the 3' end, we can map exactly where polymerase was transcribing when we did this run-on. And there are some nice features of this that we particularly like that I just want to go through quickly so that you understand what the data that we get out looks like. The first is that you get stranded transcription. And so both transcription from promoters will initiate in both directions, but it's only the sense transcript, so the mRNA protein coding transcript, where you get stable elongation. And so when I show data, you'll often see signal on both up and down. One is the sense transcript being transcribed, and the other is the antisense. In addition, this assay can pick up all RNA polymerase transcription. That's Pol1, Pol2, Pol3. It also includes non-coding transcripts, which for the purposes of studying a transcription factor turns out to be incredibly valuable, because in one assay, we can see changes in the transcription and production of the mRNA, but we also see changes in production of these non-coding transcripts called enhancer RNAs. And work from numerous groups have shown that enhancer RNA transcription highly correlates with the relative activity level of a given enhancer. And so eRNAs, they're transcribed just like transcription initiates, just like at a promoter, where you get bidirectional initiation of transcription at these enhancer regions. And we can quantify changes in those eRNA transcripts to quantify changes in enhancer activity. And then finally, probably not relevant for our talk on RUNX1 today, but it gives us also information about RNA, a little bit more mechanistic information about RNA Pol2 dynamics. So we can see the pause polymerase and we can versus the elongating polymerase. Okay, so here's an example of a gene using this approach with rapid RUNX1 degradation followed by in combination with ProSeq, where we can see a gene Wnt11 that within even 1 hour of adding this small molecule to eliminate RUNX1 protein levels, we see a significant reduction in transcription, and it continues to be reduced through 2 to 4 hours. We also see this enhancer. So these are these enhancer RNAs I referenced. And so there's 2 neighboring enhancer elements. You see the one on the left is relatively unchanged upon RUNX1 depletion, but the one on the right, you see a reduction in enhancer RNA production that really mirrors the changes in transcription that we're seeing happen at the gene locus. And so this is what we're kind of looking for when we're looking to identify these direct and immediate effects of RUNX1 inactivation on gene expression. I just showed a couple more loci, just interesting genes from the perspective of cancer and immunology. We see NMYC is very rapidly down-regulated upon RUNX1 depletion. And IL-1 beta is also, this is now being transcribed right to left. It's this blue signal you also see is rapidly down-regulated upon RUNX1 depletion. Okay, so if we look globally at what's happening, we see that we see evidence of both up and down regulation of gene transcription upon RUNX1 inactivation. The majority of genes are downregulated, which is consistent with the role for RUNX1 in activating gene expression, but there are instances where genes also become upregulated, which suggests RUNX was functioning to keep these genes in a more lowly transcribed, silenced, repressed state. In addition to this, we see, just looking 1, 2, and 4 hours, we see different kind of patterns of regulation, and that we see some genes change early and stably. So at early time points, they go off and they stay off through the time course. There's others that take longer to change, where we don't see any significant changes until 4 hours. And then there are some that immediately change following RUNX depletion, but then rebound by the end of our time course. Now, definitely the early and stable downregulated genes are the ones where we see the most robust changes, but we do also, again, see more subtle changes in gene expression, suggesting RUNX1 can also function in repression. If we do gene ontology and ask what these genes are, the activated gene targets are the only ones that have significant gene ontology signatures coming out that are present in each of the 4 time points, and those are associated with platelet activation and hematopoietic cell lineage. So again, consistent with what we know to be important functions of RUNX1. And I just want to show an example of both activation and repression side by side. So CD244 is a gene target, again, being transcribed right to left, so it's shown in blue. That is activated by RUNX1. We see a rapid reduction in transcription following RUNX1 depletion. CD226, also being transcribed right to left, is a repressed target, and we see by 2 hours following RUNX1 depletion, we begin to see an increase in transcription across this locus. Now, both of these encode cell surface proteins, so we can look at, use flow cytometry to see if the protein levels are also changed. And you can see for CD244 that depletion of RUNX1, shown in blue, results in a significant reduction in the CD244 protein at the cell surface. In contrast, CD226, while much more subtle, we do see a significant and reproducible increase in CD226 levels at the cell surface. So least examples in this case that these transcriptional changes we're seeing do in fact alter protein levels. Now, we thought these cell surface proteins might provide an opportunity as a biomarker or reporter for RUNX1 transcriptional activity to just do some experiments to see, can we look at different RUNX1 mutants and assess how they affect RUNX1 transactivation, particularly CD244, where the signal to noise was quite good. And so in red, what you can see is vector control transduced cells that have relatively high CD244. When we degrade the endogenous RUNX1 shown in blue, that level goes down, and we can complement that reduction in CD244 levels by re-expressing a wild type RUNX1 protein. So we asked, could different RUNX1 mutants complement this defect in CD244 transactivation? And what we see is really the point mutants present in the runt homology domain, which facilitates DNA binding, were the most abrogated in their ability to maintain CD244 protein levels, but they were impacted to varying degrees. So R166Q was probably the most compromised in its ability to maintain CD244 levels, whereas S141L, while it couldn't maintain CD244 to the same degree as wild type, was less compromised than R166Q. And we can then go on to use this PMA-induced differentiation to show that that transactivation activity actually pretty closely mirrored the degree to which these different mutants were impacted with regards to cell differentiation. So again, R166Q seemed to be the most affected. And if we compare that to the RUNX1 knockout, they're quite similarly defective for differentiation towards that CD41, 42 double positive cell state. Whereas S141L, that was less affected in its ability to drive CD244 levels, was also definitely deficient in differentiation, but not to the same degree as complete loss of RUNX1. So just to quickly summarize this part, using this model of rapid RUNX1 depletion, we can show that growth and differentiation are affected. We see roles for rungs both in gene activation and gene repression, and they're kind of complex patterns of regulation, where some genes change very quickly and stably, whereas others take longer to change. We think direct targets have the potential to provide biomarkers for transcriptional output, and in the context of just the few point mutations that we looked at within the runt homology domain, they have subtle differences in both the degree to which transcription is impaired, and this seems to correlate with how the effect of these mutations on differentiation. So now I want to kind of dig in a little deeper to how we figure out, once we've identified these RUNX1 gene targets, how we figure out how they're regulating gene expression. And before I do that, I just want to talk a little bit about enhancer biology and what we know are correlates with enhancer activity. So RUNX1 is one of these master transcription factors or lineage transcription factors, and we know it functions at enhancers, which can often be quite long distances away from the promoters that these factors are regulating, and does provide some, hurdles when we're doing our informatic analysis. And so we think that through some sort of either looping or condensate formation, there's actually three-dimensional changes that allow these enhancers to be in relatively close proximity with the promoters in which they're regulating. We already talked about how enhancers and promoters are both transcribed bidirectionally, so it's a common feature of both elements. And also, there's certain histone marks that tend to correlate with enhancers and promoters that will come up a bit later. So both active enhancers and promoters tend to have high H3K27 acetylation. Again, active enhancers have a high eRNA synthesis. And then there's important protein contacts that are typically made by these transcription factors, such as through mediator, perhaps cohesin or YY1, that tend to bridge this three-dimensional structure to facilitate transcription from the promoter. And so we just first started out just simply asking, where does RUNX1 bind to regulate gene expression, and how does it impact enhancer function? So we performed an assay called cut and run using the HA tag that we'd integrated into the endogenous RUNX1 locus to define where across the genome it bound. Here we're just showing the regions that it bound that reached our pretty stringent statistical cutoff for being lost up within 2 hours of RUNX1 degradation. And so that left us with about 13,000 high confidence RUNX1 binding sites throughout the genome. If we then annotate that to ask, are there nearby genes that change when we lose RUNX binding from these locations, what we see is only about 14% of RUNX binding sites could be annotated to a regulated change in gene expression upon RUNX1 degradation. And this is actually not surprising at all. This is something that we've seen time and time again when we do these studies, that these transcription factors tend to bind tens of thousands of sites throughout the genome, but we can only assign a functional consequence of their removal at a very small minority of those binding sites. And so in the context of RUNX, 86% of the sites we see it binding, but we can't, using functional filters, can't assign any distinct effects on gene expression outputs. So then we looked at features of enhancers. So they're nucleosome free. And again, we're looking at enhancer RNA transcription as a metric for enhancer activity that might correlate with this 14% of RUNX1 binding sites that seem to elicit changes in gene expression. And so here we're just looking at the CSF1 locus. As you can see, it's transcribed left to right. This dark signal on top, that's significantly reduced even within an hour of RUNX1 degradation. Upstream, there's this enhancer cluster with multiple accessible regions, which is bound by RUNX1. And what you can see is when you degrade RUNX1, there's actually a rapid loss in chromatin accessibility at specific locations. Not all of these accessible regions are altered, but very specific ones rapidly lose accessibility, and that correlates with a loss of eRNA transcription, which is, again, a readout for relative enhancer activity. And so we looked more broadly to ask how many at RUNX1 binding sites, these roughly 13,000, how many do we see changes in either chromatin accessibility or enhancer RNA transcription? And we saw many fewer changes in enhancer RNA transcription than changes in accessibility, I would say. But we asked which of these two features of enhancers more likely correlate with changes in gene expression. So is it really the loss of accessibility that's driving the changes in gene expression, or is it the loss of eRNA production? And what we see is that it's really eRNA transcription that highly correlates with changes, that significantly correlates with changes in gene expression. And with these losses in accessibility, only seem to significantly correlate when they also occur with a change in enhancer RNA synthesis. If we ask what types of motifs are present in these different distinct clusters of RUNX1 binding regions, we see that changes in accessibility actually are very highly enriched for the RUNX1 sequence motif. But at the eRNAs, we really only see eRNA changes. We see both RUNX motifs, GATA, and ETV, or ETS factor motifs. We can do the same thing for the repressed targets. I will say gains in accessibility were much less frequent than losses in accessibility. And at these repressed enhancers, it was really changes in eRNA synthesis that correlated with changes in gene expression. Again, not really any change in accessibility. But what was striking is that if you look at these different RUNX1 bound enhancers that show either changes in accessibility or eRNA production, there's a complete lack of RUNX1 motif, and really exclusively GATA motifs are being called in these regions. If we kind of dig in more to these changes in accessibility, which were intriguing because they were clearly happening, but again, not really correlating with gene expression, what we observed was the losses in accessibility not only were enriched for RUNX motifs, they were often enriched for multiple RUNX1 motifs. Whereas these gains in accessibility, again, were largely devoid of RUNX1 motifs. And then moreover, if you look at the distance between motifs in these regions, if you look at all motifs throughout the genome and the average distance between two RUNX1 motifs, those regions that lost accessibility, the motifs tended to be significantly closer together. And in contrast, of the few increases in accessibility where there was a RUNX motif, it tended to be a larger distance from the nearest neighboring RUNX motif. So we're at this point, the changes in accessibility were really perplexing us because they didn't seem to correlate with changes in gene expression. And we wanted to know, what are these regions and what can we find out about these regions? And again, it's actually quite profound and significant, the numbers of changes in accessibility that we see very quickly following RUNX1 degradation. But we were surprised that many of the losses in accessibility actually didn't seem to occur at regions that we could see RUNX1 binding by cut and run. But ATAC is nice in that you can use something called motif footprinting to actually determine, is there a protein sitting there that would obscure the transposase from integrating at these specific sequence motifs. And so we use the ATAC, we mined the ATAC data more closely to do motif footprinting. And actually, when doing that, the ATAC predicted that something was sitting at a RUNX motif, an additional 4,400 sites, and preventing the transposase from integrating. And indeed, if we plot the cut and run signal around those ATAC called protected motifs, we do see signal. It's much reduced compared to the threshold cutoff that we typically use during peak calling. And it has this very distinct pattern that it seems that the RUNX is binding kind of right at the periphery of these really narrow ATAC peaks. And so if we compare, again, these accessible regions where we see changes in eRNA, these are the regions that we know are regulating gene expression. Those that we don't see clear evidence of gene regulation, they're relatively lowly transcribed, they have consistently less H3K4 trimethylation, and they have an enrichment of this mark associated with transcriptional repression, H3K27 trimethyl. And so I would say this is somewhat unsatisfying. We have hypotheses as to what these might be. It's possible that these are poised enhancers, so that RUNX is really establishing these enhancers for, to rewire gene networks at the next cell differentiation state. It's also possible that these are something, you know, completely different, are doing something completely independent of transcription, and there's a couple of people in the lab pursuing this. So again, at genes, at enhancers that are activated by RUNX, enhancer RNA is the best correlate with gene activation. And we see profound changes in accessibility, but we can't necessarily define most or identify how that is potentially impacting gene expression. And this raises this point that's a caveat of using these rapid degradation approaches, is that what they're really allowing us to address is, are these factors required for the maintenance of gene expression? They can't as effectively address whether or not these are required for the establishment of gene expression. And so there are ongoing studies in the lab to try to understand, are these other accessible regions that are dependent on RUNX to maintain that accessibility potentially more involved in that establishment of this next gene signature that will be required for subsequent differentiation. Again, RUNX and ETS motifs are the most highly enriched at these activated enhancers, but it's very profound that there's a high enrichment of RUNX motif at these accessibility changes that we see. In contrast with repression, only enhancer RNA changes correlate, and we have much less evidence that this repression is mediated by direct RUNX1 DNA binding, because again, we see no enrichment really of the RUNX1 motifs at these enhancers that seem to be regulated following RUNX depletion. And so we're interested in what accounts for these disparate effects on gene expression. And so to get at this, we asked, what proteins does RUNX1 associate with? So we modified our CRISPR engineering approach to now, instead of integrating this degron tag, integrate a 3x flag tag that allows us to pull down the endogenous RUNX protein and use flag peptide-based elution to then do mass spec and identify RUNX1 associated protein complexes. And so we had a very robust data set, I think. We see RUNX associating with histone modifying enzymes that are involved both in activation and repression, consistent with, you know, our ProSeq data. But it was interesting that it seemed that complexes such as the HDAC complexes, co-res complexes that are really associated with repression amongst the histone modifiers were quite overrepresented. But we see, you know, almost entire complexes coming out for mediator, which we know is important for communication with the promoter and PIC assembly, with integrator, which we know, in addition to its role at regulating mRNA processing, is critical for enhancer RNA processing, and a number of chromatin remodelers, which again are going to be crucial for regulating chromatin structure and potentially impacting chromatin accessibility. I do want to point out, because I get asked this a lot, do we see B-core? We did get a few peptides for B-core in 2 of our 3 replicates. It did not reach the statistical cutoff to be seen as significant. We did get other polycomb complexes, so other PRC1 complexes. So we get PCGF5, which is kind of a defining member of the PRC 1.5 complex, which is consistent with what, you know, Alan Cantor had reported previously. So again, I wouldn't overinterpret a negative result. And if I have time at the end, I'll come back to this this comment about B-core. But we do see some peptides, but we don't see any of the other members of the PRC 1.1 complex that are specific to that complex. So no PCGF1, no KDM2B in this experiment. Now we also saw, and I wanted to talk about this because I think this was in my early career proposal, we also saw some proteins that you wouldn't normally associate with gene control and transcriptional regulation. And one of them that was most intriguing to us was this FANC-I protein, so Fanconi anemia protein. But we also see, for instance, 4 out of the 5 components of the replication factor C complex. So we wanted to ask, given, you know, we know there's this link between RUNX1 and DNA damage, and given the role in Fanconi anemia proteins at repairing interstrand crosslinks, we treat cells with mitomycin C, could we see co-localization of RUNX1 at FANC foci? And to make a long story short, the answer is we never really saw anything super compelling to suggest that RUNX was relocating to these FANC D2 foci or to DNA damage broadly. And so this kind of, we're not really sure what the association with FANC-I means at this point in time. And we'll leave it at that. I think there's still a lot more work to be done here. But what we were really excited about, given that the different motifs that were coming out of our RUNX1 regulated enhancers, was that RUNX1 regulates with a variety of other sequence-specific transcription factors. And, you know, before I kind of jump into what we really want to focus on today, I mean, some of those transcription factors, or this basic helix family of transcription factors that includes E proteins and tau1, this is very interesting to us because we also saw a significant enrichment of known repressors of these E protein complexes, which include MTG16, MTGR1, and RUNX1T1 or ETO, which again, MTG16 and ETO are frequent translocation partners of RUNX1. And so while not particularly relevant to what I'm about to talk about, this is something that really has caught our attention and intrigues us that, you know, the wild type RUNX protein seems to naturally associate with some of these co-repressors to which it's frequently translocated. But what we do see is 6 out of 7 members of this previously published transcription factor heptad that RUNX was reported to function in. And so this came out of the Gherkin's lab, and RUNX1 was proposed to, based on ChIP-seq studies, to kind of co-localize on chromatin more frequently than you would expect by chance with this heptad of other transcription factors, including FLI1, TAL1, LIL1, LMO2, and GATA. And we see all of these. The only one we don't see of this heptad is ERG. And so we wanted to kind of, you know, first see, can we see these factors co-localizing with RUNX1 around RUNX1 bound enhancers? And we do. We don't see that degradation of RUNX results in global changes in the binding of any of these factors, but we see clear evidence that FLI1 seems to broadly co-localize with RUNX1 on chromatin. For some of the other factors, such as GATA2, it was noticeably absent from the most robust RUNX1 binding sites and then enriched at some of the weaker RUNX1 binding sites. And so we wanted to go on and really kind of determine, are these other sequence-specific factors and associated co-regulatory complexes really impinging on the RUNX1 gene network? And, you know, in this cell stem cell paper where, you know, this heptad was described, in the discussion, I think they said something that was very prescient. And I put the quote up here because they said, single protein ChIP-seq studies reporting long lists of binding peaks often provide only limited scientific insights because there are far too many putative candidate target genes to provide new specific biological functions. The challenge, therefore, remains to develop widely applicable strategies for extracting biological meaning from these genome-wide ChIP-seq data. And what I hope that I've showed you in the first part is this is what we think these degron technologies are allowing us to do. We see binding at tens of thousands of locations, but we only see changes in functional metrics for enhancer activity like accessibility, like eRNA transcription at a limited number of those. And so we think that this, using this technology can help us revisit this idea of transcription factor cooperativity to really get at whether or not these sequence-specific transcription factors that we can see associating with RUNX through mass spec, we see co-localizing on chromatin through cut and run, do they actually contribute to the regulation of the RUNX1 gene network? Okay, so we add degron tags to, you know, multiple of these RUNX1 interacting factors. I think I need to speed up a little on time, so I'm going to just zip through this. The addition of the tag doesn't affect global gene expression. We can work out conditions to eliminate the protein within 2 hours, and then we can look quickly. We screened by flow cytometry for what we knew to be, again, these 2 RUNX1 regulated targets to find the ones with the most evidence that they might be kind of impinging on this RUNX gene signature. And long story short, I will say the ones that looked most promising were FLI1, GATA1, and MTG16. we included TLE3 because it's a Groucho-related family member that we know directly associates with the C-terminus of RUNX. So we perform ProSeq after 2 hours of degradation of each of these factors. And what we see is at least one of these 4 factors contributes to regulation of 58% of the RUNX1 target genes. FLY1 overwhelmingly was the most likely to co-regulate a RUNX1 target, and actually 30% of RUNX1 target genes were co-regulated by FLY, and then 13% were regulated by multiple of these other factors. So this is kind of a lot to look at, but basically each of these dots just represents a RUNX1 target gene that is co-regulated by one of these factors. If they're cooperatively downregulated, they're blue, shown in blue. So if you see a blue dot here, this was downregulated upon RUNX1 depletion and also downregulated upon FLY depletion. Red are cooperative upregulation events, and yellow are antagonistic events. So RUNX upregulated and perhaps FLY resulted in downregulation. And then the grays, there are multiple factors converging on these genes, and the regulation became much more complex. And so what we see when we kind of add up whether or not these factors are functioning cooperatively or antagonistically to RUNX1 function, really FLY1 is the factor that stands out as being most likely to cooperatively regulate the expression of a gene, whereas all of the other factors, a greater percent of the time, seem to oppose RUNX function in their regulation of the gene. Here is just an example of the only gene in ALAS2, which is involved in heme biosynthesis, that was regulated by all of these factors, and perhaps not surprisingly, its transcription goes up with degradation of both FLY and RUNX. It goes down with degradation of GATA, and it goes up with the degradation of the co-repressor protein MTG16. We then wanted to ask not just the genes, do the genes change, but do the enhancers themselves change? So are these factors actually both regulating the same gene, but doing so from the same enhancer? And what we found here was that by and large, FLY1 and RUNX1 activate gene expression from the same, so they affect the enhancer RNA transcription, so function from the same enhancer more often than not to promote that cooperative gene regulation. What was surprising was GATA seemed to stand out as being the only factor that seemed different from the gene regulation patterns, and that if GATA is at the same enhancer as RUNX, it seems more likely to function cooperatively, which raises the question, perhaps some of these shared RUNX GATA targets, they're not always contacting from the same exact enhancer elements, but there's still more to do here. And this is just one example where there's integrin alpha-2 that is both activated by RUNX1, shown in blue. So when you degrade RUNX1, transcription goes down. Similarly, to degrade FLY1, transcription goes down. And there's this intronic enhancer element with this antisense eRNA that is reduced both upon RUNX1 depletion and upon FLY1 depletion. So to just summarize, RUNX1 associated transcription factors, they do significantly contribute to the regulation of the RUNX1 gene network. And there have been other studies recently that have started to question how much transcription factors really cooperate or antagonize one another versus regulating independent gene networks. And I think certainly it seems clear that these factors do impinge on the RUNX1 gene networks. The effects are going to be target specific, and they include both cooperative and antagonistic interactions, which again is something that ChIP-seq can't give you. It can tell you it's sitting there, but it can't tell you whether it's promoting activation or repression of the gene. And then generally speaking, FLY1 has the broadest overlap with the RUNX1 gene signature, and it's most likely to act cooperatively, where GATA1 is more likely to act antagonistically. But again, we can find exceptions to every rule or generalization that we try to make. Okay, so if I just have 2 minutes, I'll just touch on what we're thinking, some ongoing studies in the lab and what we're thinking moving forward. And one of the biggest questions for every time that we've done these transcription factor analyses is really, what accounts for regulatory binding? And I think the story that I showed you today with RUNX suggests that other transcription factors may be playing a role into what the ultimate outcome is of a transcription factor binding event. Data that I didn't show, there's clear evidence that chromatin structure can certainly play a role. So there are certain chromatin environments that a transcription factor may not necessarily be able to overcome. See. Sorry, I just lost my screen. Okay, so we're very interested in, all of this was done in a cell line. We know the caveats of using a cell line. And so we're very interested in how cell type specific gene regulation can be altered in primary cells. So the same transcription factor, different chromatin state, different repertoire of co-expressed transcription factors. How do the gene networks potentially change? We're interested in kind of the interplay between transcription factor binding affinity and transcription factor dosage, which is of course important in a situation where you have transcription factors that are associated with haploinsufficiency. And we're also interested in some of these slower, more stable mechanisms of regulation that, again, these rapid degradation approaches may be missed. And can we distinguish between the role of RUNX1 and, say, establishing a gene expression network versus maintaining it? And so we also have people in the lab working on this question. And then finally, as many people are, we're interested in whether there's kind of a molecular basis for some of these co-occurring mutations that affect other gene expression regulators. And so there's an ongoing project in the lab looking at this. And so I do just want to point out, because again, I always get asked this, and we didn't really see compelling evidence for RUNX associating with B-core in our RUNX1 mass spec. So Ariel in the lab has tagged B-core now. So we're coming at this from a B-core specific angle as opposed to the RUNX1. And he does mass spec following pull down of B-core, and he sees all the other components of the PRC 1.1 non-canonical polycomb complex. But what was interesting is there were very few peptides mapping to transcription factors, but at the very top of the list were RUNX1 and CBF beta. And then the transcription factors below those were all the ones we just finished talking about, Fly1, Tau1, that we know associate with RUNX1. And so what Ariel's done now is he's made, more and more of these degron tags are being developed. And so he's made an isogenic cell model in which RUNX has one degron tag and B-core has a second degron tag. And we can now use two different small molecules to toggle RUNX1 levels and B-core levels and ask, how do they impact gene expression? And is there any molecular intersection that might account for the frequent co-mutation? And it's early days, and I don't want to get too far out in front, but I think he's seeing some very interesting things. So I'll just skip this and go to the end. And I have to thank my lab at Einstein. Joyita Chakraborty is a postdoc who did much of the RUNX1 work that we've talked about, but almost everyone in the lab has some hand on some sort of RUNX project at this point in time. I have fantastic collaborators and colleagues at Einstein. It's a highly collaborative and interactive place, and I'm very grateful for them, as well as the flow cytometry computational genomics core at Einstein. And all of this kind of came out of work that I had started with Scott Hebert when I was training with him at Vanderbilt University. I obviously have to thank the funding, first and foremost, the RUNX1 research program. I showed this picture in the past, but that is the before and after RRP came into our lives, my lab just, getting started at Einstein and being essentially empty. And, this funding and the work really provided the foundation for our recent R01. So we'll be forever grateful to the support that we've received from RRP. And with that, I'm happy to take any questions.
Thank you so much, Kristy. Wow, that is just a ton of work that you've accomplished, you and your group. We have some questions. I know we're at the top of the hour, but if you're, hopefully people can stay for a few minutes so we can answer some of these questions that came through. But thank you. What a great presentation. So the first comment is from Lucio Castilla. He says, great work, Kristy. And he's asking, can you use RNA-seq data to ask if other functional pathways besides differentiation can be defined after RUNX1 degradation, such as pathways associated with DNA damage, cell death, or cell cycle?
Yeah, I mean, we certainly can. And I think as our cell cycle data alluded to, sorry, got a little scratch in my throat.
Okay.
That actually is, I mean, we do have RNA-seq data. I don't know that we've gone out far enough to look for some of these things because I think these are definitely phenotypes, but we don't see them quite so early as, say, the ProSeq. And so I think we have RNA-seq out through perhaps 24 hours. I think we probably need to go longer to start seeing some of these phenotypes. There's clearly phenotypes happening, again, with regards to cell cycle, whether that's associated with DNA damage, I don't know whether at this point, because again, I do have my own bias that there are lots of great models to look at those things, and I don't think it's a strength of our model. So I will say that. Think the strength of our model is the stuff that happens early, and I think other models are better suited to identify some of these other pathways that happen later. But it's definitely something that we can do.
Great. The next question is from Sophie Calloway, and she also says, great work, Kristy. She said, sorry, I missed this, but what is the cell population that you're working in? And are you able to model, for example, how these changes may interact with others during development, such as GATA switching?
So we can't look at GATA switching. So this is a weird model in which, these are human erythroleukemia cells. It's a weird model because both GATA1 and 2 are both expressed in this cell line at the same time. So we can see both of them being expressed. We see both of them associating with RUNX1. I will say we have other models coming down the pipeline to ask those sorts of questions. I think cell lines for us was the easiest point of entry because these CRISPR editing approaches are relatively inefficient in the homology-directed repair. But this is what Victor in the lab is working on, and is working with models where we can actually look at different cell states in a more normal situation and push differentiation in different directions, and then remove runks from these different populations and kind of get to more of these questions, because I think there's a number of interesting questions that just require us to kind of move to some more sophisticated models, which we're in the process of doing, and I'm hoping that will shed light on these types of issues.
That's great. That was one of my questions, is whether some of the transcription factor, sort of cooperating transcription factor partners are also a reflection of the cell type that you're, the original cell,
Yeah.
right? And that
Yeah.
what might be happening in a stem cell or, a myeloid progenitor could be different.
Yeah. And I think that's absolutely right. I think we're thinking about there's going to be cell type specific networks being regulated, and we think the chromatin landscape is going to impact that. We think what other transcription factors are expressed is probably going to impact that. And we think how highly RUNX is expressed is going to impact that, which is why we're super interested in some of the work that is in the early days, looking again at dosage and binding stability and kinetics, and how that fits into which of these binding regions actually elicit a functional change in gene expression. So I think all of these things are going to factor in, and we are working towards it, but it'll take a bit of time to get some of these, again, better models, I think, that we're kind of graduating from the initial cell lines now.
That's exciting. And now we've got another question on, and I think this should probably be our last since we're 5 minutes over,
Okay.
but great talk. Given the deep profiling of chromatin complexes and gene expression changes, has this led to any potential therapeutic interventions to counter RUNX1 mutation?
This is a good question. We haven't, so the approach that we've been kind of throwing about, we haven't actually started, is performing targeted CRISPR screens based on the CD244 biomarker that I showed and the hits that came out of the mass spec. So for instance, if you have, and I didn't talk about this, but we do have models where you can delete half of RUNX versus all of RUNX, and you do see an intermediate effect on CD244. So could you perform a targeted CRISPR screen looking at these associated protein complexes to find some that, say, might restore more normal transcriptional outputs. And this is how we're thinking about it. I don't have anyone in the lab to do it at this point in time, but it's kind of one of these on the list of things to do, because I think that's going to be the ultimate goal. And I think there's lots of interesting potential targets. We can certainly go on a target by target approach, just what has a drug, and start throwing it on, and we could do it that way. But I think the CRISPR kind of targeted CRISPR screen approach is going to be the most comprehensive.
Yeah. Well, and I think it was great to see that you're starting to investigate the RUNX1B core cooperativity. And I think that hopefully will also, help identify vulnerabilities and understand how those two mutations could be changing the chromatin landscape, et cetera. That would drive progression of the disease as well. Thank you. Thank you very much for
Thank you for having me.
Presentation and all the work that you've done, and congrats. Looking forward to seeing this in a paper very soon.
All right. Thank you.
All right. Bye-bye. Thank you, everyone.